Hey guys, my name's Kat.
I'm Jack.
I'm Edie.
I'm Tristan.
And we're here talking about the Nobel-winning prize, CRISPR. It won the Nobel Prize in 2020, and we're just going to start talking about the history, and we'll go on from there. So I'm going to send you right over to Tristan.
Perfect. So CRISPR, that CRISPR stands for clustered regularly interspaced short palindromic repeats, are well-known acquired immunity systems found in archaea and bacteria. CRISPR nucleases are currently widely regarded as the most trustworthy genome editing and modification tools. The first evidence of the gene's existence was discovered in 1987 during a research of the phosphate metabolism genes, when a distinctive repeating DNA sequence, subsequently termed as CRISPR, was discovered in the E. coli genome. Equivalent sequence patterns were later discovered in a range of bacteria and halophilic archaea, indicating that such evolutionary conserved clusters of repeating sequences perform an important role.
What's kind of interesting is that the guy named Francisco Mojica was the first person to really name CRISPR locus, and that was in 1993. He basically worked on it from 1990 to the 2000s, and he reported the disparate, repeat sequences, and which is now known as hallmarks of CRISPR sequences. And he worked with a guy named Ruud Janssen, who first used the term CRISPR in 2002. And then a few years later, in 2005, they reported these sequences match snippets from the genomes of bacteriophages. This led him to correctly hypothesize that CRISPR is actually an adaptive immune system. However, he wasn't the only one to come up with this conclusion. They found another group that found similar outcomes around the same time that he did. And Jax over here has a little bit about the 2005-2006 advances for this type of research, and he'll go now.
We actually have two researchers that made some significant advancements during the time of 05 and 06. First, we have Alexander Bulletin, and he was studying the bacteria Streptococcus thermophilus and the subsequent sequencing of that bacteria. He found that the CRISPR array in there was similar to the more widely known version, but lacked certain genes and contained others that were not found within that model. Later on in 06, Eugene Koonin proposed that the CRISPR could be a cascade bacterial immune system that inserts phage DNA into the bacterial DNA. He also hypothesized that this could be used as a possible DNA repair system.
The 2010s led to fully developing the way that the CRISPR-Cas9 system functions. Sylvain Moineau and his team at the University of Laval started to track down this path in 2010, noting that it always creates double-stranded breaks in DNA at target locations, and that Cas9 is the only required protein in order to make these breaks, which is a distinguishing feature of the Type 2 CRISPR system, which involves the usage of a single large protein, Cas9. Emmanuelle Charpentier and her team in 2011 found the final piece of the CRISPR-Cas9 system puzzle. They discovered that a small RNA, which they called a trans-activating CRISPR RNA, or tracer RNA, is responsible for guiding Cas9 to its targets. In 2012, Virginijus Sixnus found that using this understanding, the CRISPR-Cas9 system could be used to its fullest extent by reprogramming the RNA in the system to target specific sections of DNA, allowing for virtually infinite possibilities of genetic editing. This was confirmed by later studies done by Charpentier and Jennifer Doudna, which won them a Nobel Prize in 2020. Dubbed by some as the year of CRISPR.
Perfect. And I would like to add, so the first commercialization of CRISPR-Cas9 technology was in April 2012, as well as the first patent submitted for CRISPR technology was in May 2012. So the guy who submitted the patent was named Feng Zheng. He revolutionized the creation of instruments for the genome editing from natural microbial CRISPR-Cas9 systems for application in eukaryotic cells, including human cells. He and his colleagues have converted many different CRISPR systems, including RNA targeting, CRISPR-Cas13 systems, and CRISPR-associated transposon systems. They can be utilized for gene insertion.
What's crazy about this is the fact that they've been working on this for 20, 30 years now, and it's just now won the Nobel Prize. If you think about it, you put it into perspective, it takes such a long time for research to be able to even think about being used in the real world. And even though we have gotten this much about CRISPR, and we know that CRISPR could potentially help and cure a bunch of diseases that humans have, we still are nowhere near actually using them in a real world setting.
The wildest thing about that, too, is that the patent wasn't even for CRISPR being used in human genes specifically. That's an evolving field that we can talk about next. And to fully understand how and why CRISPR has been used to alter human genetics, though, we should first take a brief look back at previous instances of human genetic editing. In 1988, the United States Congress funded the Human Genome Project, which aimed to completely map out the human genome. This was completed in 2003 and covered around 92% of the human genome, a huge undertaking that fundamentally altered the way the understanding of how the human genome is constructed. Partially due to this new knowledge, the early 2000s were host to many new developments in this specific field, such as the drug Gleevec, which was a targeted gene therapy drug that aimed to treat myelogenous leukemia, and Gardasil, which came about in 2006 and is a preventative cancer vaccine that was discovered using gene editing. To date, this HPV vaccine is still the only preventative cancer vaccine. After the discovery of CRISPR as a tool in 2012, individuals were quick to take advantage of its power as a way to alter human genetic material. In 2015, CRISPR was used to edit human embryos by Jinju Huang at the Sun Yat-sen University in Guangzhou. Because Huang had altered the cells that affect heredity, his experiment aiming to fix a gene error causing a blood disease was not considered ethical. It became a controversy almost immediately. This was 3 years before human trials for CRISPR were officially approved by any governing body. In 2018, human trials for CRISPR were approved. This has led to a number of ongoing experiments in the field of genetic engineering, including a way to potentially treat sickle cell anemia, as well as the editing of human embryos in order to influence its genetic makeup. In a moment, we'll take a look at these two instances of CRISPR being used to alter genetics, but first I wanted to ask the rest of you, is there a distinction, do you think, between using drugs that target genes, such as how Gleevec slows or stops cancer cells from growing, versus using CRISPR to directly alter human cells? This might be something we get into as we go further into it, but I just kind of wanted to know your thoughts.
I think the only distinction would be like the availability of the medicine rather than the actual like human doctor going in and manually changing those genes, and how much it would cost as well. Because like if you think about it, like not a lot of people can pay for all these expensive surgeries, procedures, stuff like that. So I think that's a very big thing to touch on.
I think functionally, though, it's the same. I mean, you're doing the same job either way. It may be cheaper, like you said, to do the medication as opposed to the doctor, but you're still editing the genes.
I think you could also add, like, you could say, like, if you're doing, like, using drugs, one could be more, like, instead of, like, long-term versus, like, short-term. Mm-hmm. If you use, like, the CRISPR, that could be more of a long-term repair, but if you use drugs, it could be more like a short-term fix.
Yeah, interesting points, guys. I raised the point because there is less of an ethical, I guess, concern in the modern day about drugs that are gene editing or target genes than there is from CRISPR. And I'm interested to know, how much of that is because CRISPR is this new technology. I don't know. But I think maybe looking at these two cases of CRISPR being used to edit the human genome, we might be able to get some insight.
All right, talking about the medication and like these manual procedures that you'd manually have to go in to fix, we should probably talk about a specific disease that CRISPR could potentially, quote unquote, cure. So the one that I want to talk about is just like the sickle cell. So sickle cell is a genetic blood disorder that affects hemoglobin. It causes normal shaped red blood cells to look like a sickle, which disrupts the function of transporting oxygen. This disorder reduces the lifespan of cells, which causes a shortage of red blood cells as well as obstructs blood vessels and doesn't allow circulation of blood flow to happen as it should. This also leads to severe pain and recurrent infections. These obstructions also come with clogs in the spleen, liver, lungs, and heart or eyes. Sickle cell causes an increase of organ and tissue damage and the risk of stroke occurring. So the current treatment options for patients with sickle cell anemia are receiving frequent blood transfusions or medication for pain and lower the frequencies of attacks. This is only one quote-unquote cure for sickle cell as of right now. That is to receive bone marrow from a healthy patient, but at the same time, that also has its own set of risks, such as finding a suitable donor, immune rejection of the transplant, and much more. It also is generally restricted to younger age populations because the risks increase as age increases. Now knowing that, how do y'all think CRISPR could be used to prevent or even cure sickle cell?
Well, I know in the lab that we actually just did in Dr. Schroeder's class, we talked about how CRISPR can be used to knock out a gene in a cell or in the DNA of a cell. So I wonder if something like that could be used to help either fix or change the shape of sickle cells, since that is another part of the DNA.
I personally worry about the knock-in gene therapy. I feel like it could create what's called an off-target change that might activate some cancer active cells or something like that.
And that also brings up other ethical concerns that we talked about a little bit earlier. Like I said, all this new information on CRISPR is like so new, but it's been 20, 30 years. That's like a third of somebody's life. So it's crazy to think about where we're going with this. But anyways, what we have found about the quote-unquote cure that could work for CRISPR on sickle cell is, as sickle cell anemia is caused by genetic mutation, it is a perfect candidate for CRISPR-Cas9, whereas patients undergo a procedure called gene-edited cell therapy. This is where stem cells are extracted, corrected, and replaced in the patient. Another approach would be to repair the mutation in the gene responsible for the disease and cause healthy hemoglobin to be produced. This is a gene knock-in rather than knockout. This is where the donor template containing the normal sequence of the gene is introduced so that the mutation is corrected when the cell repairs the DNA breaks with the template via HDR. And then just another approach that could potentially work is to provide a gene knockout, which is what we did in the lab, last week? Where the gene that suppresses fetal hemoglobin is switched off, replacing the mutated adult hemoglobin, potentially curing the sickle cell anemia.
Hey guys, I know that we've been talking about all the positives in CRISPR, but I'd really like to open our eyes to some of the negatives here.
Do you really think there could even be negatives? Because this could potentially cure or prevent so many different diseases that's been around for decades.
And while I understand that it can do that, there are definitely some negatives that we have to look at. To start off with, it could harm us by inducing what's called an off-target change, and I discussed this when I was talking with you earlier. This is caused by certain genes that weren't specifically being targeted by the Cas9 gene editing to be changed. Issues that could potentially arise as a result of an off-target change are the inadvertent activation of cancer-causing genes, the rearrangement of chromosomes, and the inadvertent deactivation of untargeted genes. In addition to this, there could be unintended risks associated with targeted genes. For instance, it's possible that altered genes may generate an immune response, meaning your body would attack your own cells. Even further, there's a risk that once Cas9 gene editing became widespread, we may have an issue with certain more favorable physical traits being quote-unquote picked more often, and our genetic variability as a species will severely decrease. This socially could also become an issue, as if one does not have the certain preferable traits, that person may be severely discriminated against. And there's a risk that certain social, economic, and political forces may try to force people who are deemed unfit to change the genes of their children into something more favorable. Not to mention, the term being used for human babies who have their genes edited is designer babies. And do you know how much bullying is going to go on in school for one of those designer babies?
Do you really think it would be bullying, or do you think it'd be cool? Because, personally, if I was in elementary school and knew that somebody got their genes edited, that would sound so cool. How do I do that? How can I do that?
Maybe for you it'd be nice, but for little Jimmy over there who's got brown eyes and strangly black hair, he could really use some gene editing. He's ugly. He needs help. Sorry, guys. I get a little emotional when we're talking about Jimmy. But there's another large issue with us starting to use genome modification, and that would be that we really just vetted it. Much like any medical treatment, CRISPR-Cas9 gene editing should go through many different trials and years of thorough research and development before being made available to the general population. Any early trials that go poorly would surely spread panic among the public, which will decrease further funding and support for future gene editing projects. And that's not to say that a select few should have access to it, as that poses another problem of expense. If only the rich can have access to what would presumably be a very expensive procedure that's unsupplemented by the government, then other people who cannot afford the gene modification would suffer. Not to mention, while we're on the subject of cost, that optimizing and using a protocol for Cas9 gene editing in a lab where that has not been done before is rather difficult to set up and is also very time-consuming and expensive.
So, like, Gene, like, how long do y'all think it will be until you will be able to use this on humans?
There's really no telling until it's done.
Well, that doesn't help, does it?
I mean, it was only really allowed for use on people in 2018. I mean, people obviously did things with the human genome and CRISPR before then, but the fact that it's only been 5 years, I think.
So you think we'll have, like, multiple years until this is actually able to cure quote-unquote diseases?
I think so, and I definitely think it'll be even longer before it's even really in the public consciousness.
And be available to the public as well, and not just the upper class who are able to pay for it.
Exactly. The general public doesn't even know about this technology. And when they do, when it does inevitably come into the public conscience, it's still going to be rich people who have the access to it first, to make things like designer babies, or what you were going to talk about, Tristan, right, was specifically Russia using CRISPR to potentially knock out the pain gene from cells, right?
So let me ask y'all a question real quick. So how would y'all like to remove this pain gene from your life?
I would like to not feel my ACL tear. That would be amazing.
Jack?
There's just so many issues involved with removing pain. Say I set my hand on a stove, but that stove is on, but I don't know. Now I got a hand that's on fire. What am I supposed to do with that? We need pain in our lives.
Yeah, it might be a nice idea, I don't feel pain for things, but I think in practice it's definitely not. It's just not viable.
It's wise.
I mean, it's so basically, like, in theory, it's good, but like in the long, like in like day-to-day life, like you mentioned, like your hand on the stove, you need pain to understand that. So I do agree with y'all. Like, if let's say, like, it could be if you use it in sports or even the military, where I was going with this. So let's say this got used and fell in the wrong hands and for the military. Russia has recently been talking about that and moving it for their military, ultimately making them super soldiers.
Is it bad? Is it a bad thing because Russia's doing it, or is it bad because it's just not us, or is it just ethically wrong?
Well, I think that's a loaded question. So let's take it to like let's say because it's Russia. I think if this fell into the wrong hands, the wrong country did it with their military, they could use it for like bad, morally wrong, ethically wrong causes.
But that would be for every country. If you think about it, every single country, no matter how great of a country it is, there's going to be corrupt police, military, officials, FBI.
There's always bad apples, basically what you're saying.
Even beyond the concept of bad apples, I think
every country is only working in its own interest.
Yeah, but I think even just from like a purely ethical standpoint and a purely physiological standpoint, I think removing somebody's ability to feel pain or understand that they're feeling pain is something that would just make it very hard for somebody to live a life in the everyday world. Even something as small as a splinter could lead to a huge infection if you didn't feel pain.
Imagine you got cut on your back. You wouldn't know until you bled out, passed out from blood loss.
The idea of using using CRISPR to edit the human genome in a way that would create a human for one very specific purpose, I think that's a very slippery slope into humans as commodity, which is I really don't like. So it's tricky. It's definitely tricky.
Thank you for that. Has actually changed my opinion today. Thank you.
Well, I'm so glad to hear that. I think that's the goal of every podcast creator, is to change some minds. On that note, I would love to hear everybody's final thoughts, opinions on the ethics of using CRISPR to edit the human genome. Kat, what do you think?
With certain instances, like I said, like before, curing sickle cell, quote unquote, I think it'd be great. But there's also ethical concerns, like when Tristan was talking about Russia and how they were trying to make a super soldier, And that just, I think personally it is completely wrong to do that. Jack?
I personally believe that the gene editing medications could be a good way to cure some serious diseases that we have in our society. However, I do not support the CRISPR individual gene editing system. I feel that that would be far too easily manipulated into something malicious by the higher powers in this world.
I agree with Jack on that, actually. So, I mean, if you can't be manipulating the genetic code, so that's going to go down, like what Jack mentioned earlier, it's going to be passed on generation to generation. You never know what's going to happen. It might be placed in the wrong spot. The military stuff, like making super soldiers, it just goes ethically, if it falls in the wrong hands, it could be detrimental to that.
I think CRISPR has a lot of good uses to it and good potential. Diseases like sickle cell, it would be great to have something that works every time to eradicate diseases like that from, the population. But I also think it's a pretty slippery slope into eugenics territory, to be frank. Talk of designer babies and choosing traits that we like, that's getting awfully close to, letting prejudices influence the actual next generation of children. So it's a technology, and technologies can't by themselves be inherently malicious. But I do think it's something that, especially as the years go on, I think it's something that we need to watch. Be aware of more than anything. As the general population, it's going to be up to us to decide how we want our science affecting us. So, as long as good people are out there, I think CRISPR will be used for good. But
all right, I think that's all we have for y'all today on CRISPR. Enjoy these bloopers.
Sorry about that. I really get emotional talking about Jimmy. Not to mention, there's another large issue with us starting to use genome modification.
And then in 2012, I didn't look this up. Virginigiousness? That really long one?
That's actually, that was pretty...
Is that what it is?
Don't know, but that sounded really good.
Okay.
Like, how can I do that?
Maybe for you it'd be nice, but for little Jimmy over there, who's got brown eyes and strangly black hair, he could really use some gene editing. He's ugly. He needs help. Pause it.
Thank you for that. Has actually changed my opinion today. Thank you.
Well, I am certainly glad to hear that. I think that's, the goal of the podcast. Widening the...
Widening the scope.
The scope! I couldn't think of a word!
Widening...
You didn't stop it!
No, I'm not going to.
Oh, it's still going?
Yes.
Great. Awesome. I'm so glad to hear this later when I edit it.